Thank you, Christian, for the introduction, and thank you for the invitation. I'm very happy to be here. So today I'm going to tell you a little story about phenotypic robustness. So I've been thinking about how complex traits are regulated, genetically speaking, and most of the time when we think about traits, we think about averages of things that we measure. Today I'm going to tell you a story about not the average of your trait, but the variance or the variability on your trait, and that's what I'm calling phenotypic robustness. So I probably don't need to, talk too much about this to this audience, but the conceptual framework that we use in my lab to try to understand the relationship between genotypes and phenotypes is the genotype-phenotype map. And the idea of this concept is that we are really interested in understanding how genetic variation in the shape of SNPs or whatever type of variation you measure propagates through these layers of the genotype-phenotype map to generate phenotypic variation, and how this phenotypic variation maps onto fitness. Of course, we normally try to link genetic variation to a phenotype, but as you can see in this gray, this is really weird to use. But all these layers that are gray in the middle are all the action that goes between the genotype and your phenotype, right? That you can think about development and gene expression and everything that happens to be able to translate the variation from the genome to the phenotypes. So as you, of course, all here are aware, you have different types of genotype-phenotype maps. Some of them can be very, very simple, and we can think about the beginning of biology as us trying to poke one particular gene and try to figure out if my trait is black or white, or if my fly dies or lives, right? So these are very simple genotype-phenotype maps, but all of us here included, we like to have a hard life. So we really are into complex traits, where are these type of traits that normally vary continuously in a population and are regulated by hundreds or thousands of genomic loci, right? So in my lab, we like this type of traits. And this is just to highlight that this distribution of phenotypic variation, it's found everywhere. So this is the classic example of human height, which is the textbook example of a complex trait. Okay, what is that? And for example, this is a distribution of phenotypes in flight performance of flies. You have very, very good flyers, very crappy flyers, and in general, you have a nice normal distribution. This is the distribution of longevity in fruit flies. You can have genotypes that live in average 15 days, but you can have genotypes that live 60. And even in species and traits that we have done everything we can to reduce variation, like for example, selecting crops or selecting cows for milk production, we don't manage to get rid of it, right? So you still have average good producers of milk and really, really terrible producers of milk. So in my lab, we're really interested in understanding how this variation between individuals stays, it maintains, and how it evolves. So how do we do this? Then, as Christian said, I moved from mice to flies, so I can actually get good sample sizes to do statistical genomics, which is what we do. And what we try to do to actually have a good representation of genetic and phenotypic variation is that we work with populations that have been collected all over the world. Initially by Andy Clark's lab at Cornell, he collected inbred lines from many different populations. And what we do in the lab is that we cross these inbred lines that they collected using painful round-robin crosses to try to go from the inbred line to the outbred universe of flies, where every single individual that you have, that you measure, and that you can sequence is a different genome. So this is more or less how we keep our populations, depending on the experiments, normal vials, as you know. I saw around some of these small cages for some of the experiments you guys are running. And these big cages where you can have around 100,000 flies are also something that we normally use in the lab. Okay, when we think about genotype-phenotype maps, which is how do we link genetic variation to phenotypes, most of what we know, of what I've done and what most of the field has done, usually comes from one environmental condition. Just happens to be your favorite environment in the lab, or just what we call control environment in the lab. But the question, and for this, of course, you all know this. We talked about this the whole day. We use GWAS, right? We try to use statistical inference to draw these little arrows between genomes and phenotypes. But what happens if we change environments, right? So of course, people in biology are really into environments because that's what you have to adapt to when something changes. But here I'm not thinking about populations that are already adapted to different environments. This we know, right? Like phenotypes change depending on which environment you are living in. Here, what I'm thinking is what happens if you have the same genetic pool, exactly the same, nobody's adapted to anything, and you simply expose it to 2 or to 3 or to whatever you can number of environments. So as I said, we do know that phenotypic distribution usually is affected by environments, and the fitness to phenotype map is also different. But there's a lot of still to be explored here. What happens if I do a GWAS in the blue environment, and what happens if I do a GWAS in the orange environment? Am I going to find the same number of loci regulating your trait? You're going to find different numbers. Are you going to find the same with different effect sizes? And this is what we think about as genotype by environment interactions. Okay. Another thing that we normally do when we think about these genotype-phenotype interactions is that we focus on averages, right? If you try to understand the genetic effect on a trait or the environmental effect on a trait, you normally focus on the average of your distributions. For example, if we are thinking about height, and I tell you that a population, I have a gray population and a blue population, and you measure the average of that population, and the average is the red line, there's nothing going on there, right? So we'll say, okay, this is not a trait of interest, I forget about it, and I move on to the next trait, right? This is normally what we will do. But there's other aspect of phenotypic variation, which is very interesting, that normally we don't think about, because of course it can be confounded with noise, is the variation around your average, right? So if I tell you now that they have the same mean, but the blue population has a wider spread of individual values, while the gray population is more robust, I hope we can agree that something potentially interesting might be going on in the differences of these averages, right? Again, and this is not something that I just realized it was interesting. Of course, we think a lot about dispersion in traits. If you think about, for example, plasticity, not from the point of view of changing your mean phenotype, but plasticity as the expansion of the phenotypic range that you can explore in a new environment, this is exactly a representation of that. You can think, for example, that in the new environment, for whatever reason, this tail of phenotypic values is going to be more beneficial. So the blue population is going to be more fit than the gray population. So there are so many ways in which we can imagine that variance or variability in your traits is important. Another way in which you can think about it, if you are used like me to think about regressions, is that normally what we are looking for when we are trying to understand, for example, 2 different environments, is that the mean phenotype changes, right? If we don't see this, we kind of ignore it. But you can also imagine that the change in environment, despite no change in mean, is also producing an expansion in the range of your phenotypes, right? Okay, so this idea of studying another axis of variation that is not necessarily the average value is present in many, many different fields and has been studied in many different ways. So what I will do here is just to walk you through some examples. So depending on what's your field of interest or what's the phenotype you're measuring, maybe you can relate a little bit to why this might be interesting. So if you are thinking about morphological variation, we have been thinking about developmental robustness as a proxy for variation or noise in phenotypes, and that's normally approximated by the symmetry between right and left sides of an organism, right? So you can think about every organism that is symmetrical starts from a genome, the same genome that should generate right and left sides of your body, right? So in theory, if there's no noise and everything is perfect, you should be perfectly symmetrical, right? The degree of asymmetry between right and left is a proxy for how noisy your developmental process was. So this is traditionally used in morphological studies to approximate your robustness as an individual. People who think about adaptation to new environments, for example, diet, think about this expansion in variance as another axis of plasticity. Not changing your mean, like many people think about plasticity like that way, but just how much you can explore in the new environment. If you are thinking about transcription, the field of transcriptional noise is really, really old. People have been really thinking about why do you get different variation in gene expression in cells that have the same genotype and live in the same environment, right? And they have termed this noise, but today I'm going to tell you a little bit about why we think this is not really noise, but it contains a lot of information. If you come from the quantitative genetics field, for example, this is not something that you will get taught in your basic quant-gen courses, but people, especially breeders, that have been working, for example, with chicken, with rabbits, with cows, they have been measuring the heritability of something that is called environmental variance. This means when you raise or you select some crops or some cows or some chickens, what is the dispersion of the babies you are going to have, right? So your mean is what you're trying to increase, but what can I inherit this variation around the mean? And this is really cool statistical approaches to, and we know that the environmental variance, what you could call noise, is actually heritable. So there's a way in which genetically you can explain the noise around your phenotypes. And lastly, people who study behavior, they really are into this intragenotypic variability. This means if you have a same genotype in the same environment and you measure it again and again and again, there's always this dispersion around it, but this dispersion is genotype specific, which means noise is genetically regulated. In this example that I find is pretty cool, they exposed Drosophila to these Y mazes. This is the lab of Ben de Bibort at Harvard. And what they measure is how many times a fly goes right and how many times the fly goes left, right? What do you think should be the average? 50-50, right? Or why would I go right more times than go left, right? There's no stimuli. What they find that this is true. This is genotype A, for example. The average is 50%. This is genotype B. The average is 50%. But you can see that the dispersion around it is very, very different, right? And this is done for many different inbred lines. So we do know that noise is also genetically regulated in these situations. So today, the story I'm going to tell you is about transcriptional noise and how this variation or this noise might actually matter for adaptation to new environments. So the story is about transcriptional robustness, and I hope I can walk you from genes to environment and back, telling you a little bit about what's going on in this GP map for robustness. So the first question we ask, and I think you are questioning this idea of variance. Of course, I gave you evidence that it might matter, right? But of course, how do I distinguish that something is noise? You know, like we all do experiments. We are in good mood on Monday. We are in bad mood on Tuesday. And this introduces a lot of batches and noise in our experiment that not necessarily is biological information, right? So before we thought, okay, let's go into transcriptional variance, the question was, is this important? Can I actually, many of you have worked with transcriptional data, RNA-seq here, and you know this is really noisy, right? So if you're gonna go into researching this, you better convince yourself that there's something that is not actually just noise, right? It's biology. So as I said, many people have been approaching this question from unicellular organisms that are usually not genetically variable. So they are single cell lines, for example, or they are yeast that have just one same genotype. But we thought, because we are interested in evolutionary processes, is what happens if you now go and check noise in an outbred population that has genetic variation and has all the insanity that goes into a multicellular organism. So we focus on human populations. So this is Scott and Diogo. This is work that we did in my time at Julian Aroles' lab back in Princeton, and they were the people leading this study. So the question is transcriptional variance a gene property? How do we answer this? So if it is a gene property, and I'm measuring, which I apologize because it's just hard with this pointer. This is gene expression level, and you measure gene A in context 1, let's say in my lab, and now you measure gene A in context 2, for example, Christian's lab, and we measure the same variance around the average expression of these two genes, that we will say, okay, variance is a biological property because despite the fact that we have different people and different countries, we can recover the same metric. So if this is true, and now we do a correlation between the transcriptome measure in my lab, the transcriptome measure in Christian's lab, you will, in theory, find that gene A has low variance in both environments, and gene B, for example, here is high variance in both labs, right? So this is the idea. Despite all that can go on, I can still recover this property of this particular gene. If, on the contrary, this is actual noise, and it's something technically induced, for example, I will expect that the variance of gene A in one context doesn't correlate with variance in the second context, and the overall transcriptome correlation will be a no correlation pattern, right? You could be high or low, low and high, and there's nothing that keeps you together. So how do you approach this? Of course, you need a large data set of RNA-seq data that you can overlap for most of the transcriptome. Most of this data is available, publicly available for humans, because these guys have been doing this forever. And what we did is we selected 57 RNA-seq studies that had decent sample size. So in average 250, but it ranged from I don't know, remember 30 or whatever to 1,000, right? So you have a wide range of sample sizes. After overlapping everything, we have 65% of the transcriptome that we can work with, which is not bad, right? Meaning we can actually make a decent inference of what's going on the transcriptome level. I'm happy to discuss this, but you use mean independent estimation of variance because mean and variance is correlated in RNA-seq data. And everything I'm going to tell you is based on ranks. What is most variable, what is least variable, not on the absolute variance estimation. Exactly. So what we did there is we did a rank correlation between every single pair of studies. These are 57 studies against each other. And I'm showing you here the distribution of the Spearmark correlation across every study. Long story short, we are living in this scenario where actually the variance across all these 57 studies is very consistent. We get that more than 65% of these pairwise correlations are very high. I don't know if you have ever worked with RNA expression, but in general, biological data that has higher than 50% correlation is a really, really strong correlation. If you work with RNA-seq, you will be even more surprised that this is there. So this is telling you that not the mean expression, I'm not talking about average, the variance or the noise is highly correlated and is gene level property. Yes. Yeah, exactly. This comes next. But right now, every single individual is different. Mm-hmm. No correlation. So there, it will be like this circle. Mm-hmm. Good. So now that we believe, I hope you believe me, that gene variance seems to be very consistent across genes, then we said, okay, what if anything, is regulating this amount of noise per gene? So what we did is we took the top most variable genes, so this is 5% most variable genes, and the bottom most variable genes. So genes that are very, very robust, always, and genes that are very, very variable, always. And then we started correlating this with whatever we thought will make sense. So first, we checked, because these, again, are outbred populations. These are human populations with the genetic variation. We asked, okay, which type of genes has more nucleotide or higher nucleotide diversity or has more adaptive substitutions? So which type of genetic sequence allows more mutations to live in there? What do you think? Which the most variable or the least variable genes will have more higher genetic diversity? Say? More variable, more diversity. Mm-hmm. Anybody disagrees? We're a few people. We can have a conversation if you want. Exactly. So genes that are very, the bottom of the variable genes, they have really significant less genetic variation and substitutions than the genes that are more variable. Then we ask, okay, how tolerant to mutations are these genes? So if I poke this gene, does the human die, right? So are they intolerant to these mutations? Which one do you think is more intolerant? So they don't like to be touched. Exactly. The robust ones will say, no, I'm intolerant, don't mutate me because I will cause a severe disease in this human. Then we said, okay, what about connectivity in gene networks, right? Can I use variance as a proxy for how well connected genes are in gene co-expression networks? Which one do you think will have more friends? High variable? The least variable ones. Let's see. Correct. So genes that are more stable, more reliable, and they almost always express in the same level, they have better connectivity in gene networks. And lastly, we ask, okay, there's any biology that we can capture here by the easiest analysis that you can do, which is a geoterm enrichment. Is anything here that groups all these high variable genes together, or that groups all the robust genes together? Any guesses? Which enrichments will I get for the most variable genes? Say? These guys? Basic. The less variance, basic functions? Getting information from the environment? Who gives more? You guys are very sharp. We have high variance genes or noisier genes tend to be associated with immune response or sensing of the environment, which I guess makes sense. And genes that are very robust and you really want them to be reliable, they tend to be housekeeping genes. Okay, so I do think gene expression noise is evolutionarily relevant and contains a lot of interesting information. But I told you, what about the environment? What about this GP map that I can move my genetic populations around, my genetic variation around, and try to figure out what's going on? So here is where James' story comes into place. James is a PhD student in the lab, and he took this population, one of the populations I showed you at the beginning, coming from the Netherlands. This is, again, a population that was reconstituted or made into an outbred population by the painful exercise of doing a round-robin design of every one of these lines up to the last generation. So every population here saw each other, every line. We discussed this with some of you, how painful doing these round-robin designs are, to try to make a population that looks like here. Of course, you don't get that out of a round-robin design. We do a round-robin design to make sure that every single allele that I have in my initial lines is present, and then I let this population recombine in high population sizes for around 100 generations. So this assures that what you are starting with is not going to be really affected by random drift of a haplotype taking over today or tomorrow. You actually have this, let's call it stable allele frequency distribution that has been living in the lab and has recombined for enough generations to give you low LD, small blocks, so you have high resolution when you want to map. Then what James did is he exposed this same population, again, the same genetic pool, either to control food or to high sugar food. And he collected females, in this case, age matched, try to regulate all the technical noise that we can. And he collected around 2,000 matching DNA RNA pairs. So these are 2,000 individuals with DNA and RNA data. And as Christian said, this was only possible because we developed library preparation approaches that made this actually possible. Instead of costing an impossible amount of money, now we made libraries for around 2 euros or something like that. Why this is important? Doing GWAS, of course, requires high sample sizes if you want good statistical power, but when you are mapping variance, it becomes even more important because it's statistically more demanding. So either you have large sample sizes when you want to study noise or variance, or you don't, because it's really hard to distinguish what is technical noise or randomness of your sampling and what is actual metric of variance in your distribution. So this is to show you what happens here. First, this is RNA data for high sugar and for control population flies. And I just want to highlight the fact that your transcriptome changes when you are exposed to different diets. But the most important message here is that this is the PCA of the DNA of these flies. And this is going back to what Christian mentioned. This is exactly the same pool that I actually represent in the two conditions, right? That's why these experiments have to be done very carefully, because if you don't, it might be that some genotypes prefer high sugar, and you're going to get structure when you are doing your analysis. If you get genetic structure, then it's really hard to actually tell that this is a genotype by environment interaction. It might be just genetic preference for certain diets. Good. So first, I'm going to tell you about the environmental effects on transcriptional robustness that are universal. So you might say, if it is universal, there's obviously not environmental effect. That's the message. There are things that don't change when you are exposed to a new environment. And I will show you this just not to be repetitive, but because this looks exactly like what I showed you before, but this is the flight data. So this is just to tell you that using very similar metrics, we find exactly the same that we found for humans. So this is just to say that these forces or these correlations between variants, it's kind of a universal thing. You find them in human populations, but you also find them in fly populations. Now what we did, going back to how we analyze this data, is we rank our genes from the most variable genes here to the least variable genes here, and we compare what's happening in control and in high sugar diet. So here we're looking at the top most variable genes in control is our baseline, and you can see that most of the genes remain high variance when you expose flies to high sugar. For reference, this is what happens when you compare mean expression. So this is variance or expression noise, and this is average mean expression. So, 84% of the genes that I'm showing you here as the most variable genes are actually shared by both diets. So it doesn't matter that you put me in high sugar, I will always remain high variance genes. And this is very interesting because as we saw before for humans, this correlates with response to stimuli, immune response, how you sense your environment. So this is really interesting because it means there's a need, I apologize for this terminology, to remain high variance regardless of which environment you are exposed to. But now if we take a look at the bottom, so at the genes that are most robust in theory, there's a lot of action going on. So you can see that there's all the genes that are least variable in control. Some of them are really increasing in variance in high sugar. And the other way also happens. You have genes that are very high variable that now they become low variable in high sugar. So there's also things that are happening there. We will get into that in a second. But I just want to highlight, again, the things that don't change. So 50% of this quantile here is shared between both diets. And the results were very interesting because these pathways, let's say, that remain tightly regulated or very robust are associated with development and reproduction and oocyte development. Remember, these guys are females, which is very interesting. So pathways that are making you have babies or produce the next generation, they always, doesn't matter where I put you, remain tightly regulated. Okay, but now what happens with the things? Yes. The enrichment, yes. So when you go to the middle, this is really interesting, there's a lot of fluctuations that are very small. So we think that the information contained in the middle, in terms of variance, is very noisy in the terms that you can be one quantile up, one quantile down, and it doesn't really contain much information. And this was very interesting because it was the same in humans. So in humans, we try to do enrichment for different quantiles, and it doesn't disappear completely, but you go from clear, specific enrichment in the tails of your distribution to nothing in the middle. So I think it's a general property of intermediate variance, might not really contain anything very specific in terms of biology. So what happens with things that actually are context dependent? So here I'm reminding you that the average expression level does change when you are exposed to new conditions. And this is just to show you something that is just a normal differential expression analysis. It's a volcano plot where we have around 50% of the transcriptome is differentially expressed. This is average, right? So the average expression level between high sugar and control changes. I'm just highlighting the sample size again because it's very obscene. You never use 2,000 samples to do a differential expression. Why would you do that? But I'm highlighting this here just to tell you that because of the sample size, we know that we are capturing everything you can capture. But even the tiny, tiny, tiny small effect sizes, you have statistical power to detect. And this is just important because I'm going to compare it to variance. And then I just want you to believe me that we are seeing what we can see in terms of differential expression. So these are the results for, this side works better for me. These are the results for differential variance in between control and high sugar. So these things to the right are genes that increase variance in high sugar. things to the left, they decrease variance in high sugar. The first message that you should get here is that gene expression variance increases genome-wide in the novel environment. You can see the numbers here is around 7,000 genes increase variance when you go to high sugar, while only a couple of hundreds decrease variance. So this is a genome-wide increase in variance, and we could discuss why this could be the case. So basically, we are living in this universe where control conditions have low variance relative, and high sugar have higher variance. This isn't the actual variance. This is using double hierarchical models that will first model mean changes. Again, mean and variance are correlated, so you always have to account for it. And then you map differential variance, but with the actual data, not the raw. So here you actually can get magnitude of change instead of just position in your rank. Good, but something that I find very interesting is these orange genes here are genes that change in mean between your conditions. And now I've colored my variance plot by orange, which means genes that change variance, but also change mean. But the interesting ones, I think personally, or more interesting, are the black ones, which I'm calling pure variance changes. And this is to me really interesting because when you have mean and variance changes, it's really hard to, which is the cause, what is causing the action, the change in mean or the change in variance, right? But if I tell you that all these black dots don't change their variance, don't change their mean, but they do change their variance significantly, then it's interesting because then I can argue that whatever is associated with these genes might be driven by changes in variance. That's why this obscene number of 2K here is important, because I'm very, very sure or very confident that these genes do not change mean expression. Okay, so I want to tell you a story about the genes that increase in variance. You know, the normal thing you will do, again, is you run some pathway enrichment or geo-enrichment, but who will run enrichment analysis on the whole transcriptome, right? Because here, basically, I get increasing variance in the whole transcriptome, right? And then initially, when I saw this stuff, I thought, well, this is the organism freaking out. I'm putting you in the new environment for one generation. You are kind of out of control. You don't know what will happen with you. Maybe that's all we are seeing is just dysregulation of the system. But then there's another more interesting option. And maybe we have on-off noise states of the transcriptome that are modulated by global modifiers. So it might be adaptive, and this is again just a blah, blah, blah story. We'll have to test that to increase your variance when you are exposed to a new environment. And they might be the switches for on and off noise. So this has been proposed before in different scenarios. In particular, I'm bringing the heat shock protein 90 example because it's more likely that you know about this. Heat shock proteins are chaperones. So they help other proteins fold in your cell, meaning if there's something wrong with these proteins, there's a lot of things that are going to go wrong in your system. A lot of noise is going to be there, but also a lot of exposure of genetic variation that before was neutral because chaperones took care of folding you correctly. Now, if you had a wrong mutation, all this is going to be visible, right? So this was a very famous and big topic in the early 2000s, where people showed that modifying heat shock proteins in flies would generate a lot of random and weird phenotypes, but that was also true in plants, right? So the idea, what we are going after now, is trying to figure out if we can find these switches of global modifiers that could be potentially correlated with increase in noise in our transcript. Okay, but I told you that we have a couple of hundred genes here, so I will dare to try to figure out what's going on biologically with these genes for two reasons. First, because a couple of hundreds is a reasonable thing to try to figure out biology out of that. It's not the whole transcriptome. But second, because these guys are going against the trend. The trend of your transcriptome is increased variance. And these guys are saying, no, 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 for whatever reason, I'm going to go against this. And this is harder to explain, right? Because the other one could be dysregulation, but this one is, again, apologize for the word, it's acquisition of regulation. Now you are tighter regulated, you express the same level in every single individual. So the first thing we did is, okay, let's take the orange dots, which are genes that change in mean and variance. Again, hard to say what's the causal change. But these genes who change in both aspects of gene expression are very explicitly enriched in neurogenesis, in neuron projection development. There's no other geoterm. So this is a very, very specific enrichment for these genes, which is very interesting, but again, hard to explain what is causing what. Then we go for the black genes, which are the pure variance genes. So here I know that the change is only variance. And this becomes even more interesting because they are also very specifically enriched in developmental processes, morphogenesis, and in general, metamorphosis processes. Again, I'm not selecting the 5 geoterms that I like. This is the only enrichment that these genes have. So it seems to be very specific that the genes that are really robust in high sugar conditions are associated with pathways that we believe are crucial. You better develop if you want to survive in this new environment. And this for us is very interesting because I was discussing with some of you today that when you put flies in high sugar, the metamorphosis process of the developmental timing slows down, and it takes them around 30% longer to develop. So these poor flies are dealing with something. What is it? We don't know. Something is going on when they are eating high sugar that the development slows down a lot. So 30% is like 1 or 2 days extra, right? If you are competing with a faster development population, this might really, really matter, right? But the interesting thing is that when we measure egg to adult survival as the ratio of eggs that actually become functional adults, there's no statistical difference between control and high sugar flies. So they are suffering, they are delayed, something is going on, but they manage to make as good flies, adult flies, as the control flies can do it. So they are coping with whatever is going on by making a really good fly, a really successful metamorphosis process. Of course, I don't have a way of directly proving this, but this is very suggestive that this regulation or tight regulation of these metamorphosis pathways might be related to how they are able to reduce noise and make really tight adult flies. Okay, and then the $1 million question for people who are thinking of GP maps is, what is the genetic basis of all this, right? Because I could tell you this contains a lot of biology, but it could be plastic in the strict sense that is not genetically regulated, right? So how do you answer this question? Again, using our best friend, GWAS. And this time, what you try to do is not to, this is the normal QTL mapping, or normal GWAS. You try to associate genotype with changes in the average of your phenotype, right? When you are thinking about variance QTL mapping, you try to associate changes in variance with your genotype, right? So this is just another aspect that you are checking. Instead of mean changes, now you are going for variance. Again, here, even more important than before, sample size really matters. Because again, variance when you have 3 individuals might be just a random sampling of your population. Variance when you have 500, maybe I believe it, right? So this is again a very, I have to admit, it was not trivial. I thought it would be something that we will solve in a week. And it's not, it's really computationally demanding when you are working with outbred populations that you need to account for the structure of your population. If you have unrelated individuals, it's easy. And this took a while to try to figure out how we can do it for 10,000 phenotypes, which are your genomes, in 2 different environments. So poor James suffered through this, but he managed to make it work using, again, this mapping using 2,000 individuals for which we have these DNA and RNA-seq pairs. I'm happy to discuss the details if you are interested in the technical points, but we use VEQTL mapper, which is an implementation to do variance EQTL. This is, again, as I said, for the differential expression analysis, a double hierarchical model. You map away the mean effect of your loci first, so you are not mapping variance as mean effects, and then you map the residuals of your mean mapping, and this is how you map variance. Again, this is basically computationally impossible to run with a GRM, so you cannot run mixed models. I mean, you can if you have one trait. If you have 10,000, it will take you your life. So we use an approximation where we regressed out the effect of the genetic relationship matrix, so the relationship between our individuals. And then we run this model so it becomes more efficient. Unfortunately, we also doesn't allow us to run proper GY models, which I'm very sad about. So you have to run them independently for each diet. And at the end, we are using common SNPs filtered by LD, and at the end, it's around 380,000 SNPs that we are working with. Okay, I still don't have a good slide for these results, but I'm going to walk you through it. So the answer to is transcriptional variance genetically regulated is yes, and we are very happy about this. We find that around 90% of the genes have genetic regulation for variance control, which blows our minds, but we are very happy because this means variance can be thought about as a trait, right? Instead of being just a property of your distribution, which has genetic bases that can evolve potentially. We are also kind of confident that this number is not insane because we use these same populations to map eQTLs, and we found genetic regulation for around 96% of the genes in the genome of this particular outbred population that I told you we designed to have good mapping power. But the most interesting thing to me, which I really like genotype by environment interactions, is that this genetic regulation is strongly environment dependent. So we find variance QTLs for a couple of hundred genes, but we find variance regulation for almost the whole transcriptome in high sugar, which is, again, interesting if you think about where can you map things, of course, when things vary a lot. And I told you that the variance of gene expression in high sugar increases. So I think, of course, a product that the variance, the phenotypic variance that you try to map increases. But we are also not surprised because we have done, as Christian mentioned, GWAS for longevity in control and high sugar in a different outbred population of flies, and we found that around 30% of the loci that regulate lifespan are environment dependent. So there's tons of genotype by environment interactions out there. It's just hard to map because it's hard to do the same experiment in two environments. So these results suggest that cryptic genetic variation underlies environmentally induced phenotypic variation. And this is, again, super exciting for us because, of course, cryptic genetic variation has been argued forever, theoretically, that it plays a super important role in adaptation to new environments and is critical to explain this robustness and evolvability dilemma in evolutionary biology. I don't know if somebody here is not aware of what cryptic genetic variation is, so apologize if this is too basic. But the way you think about it is if you have a phenotype here and you happen to be unlucky and you measure the effect of the red and the blue genotype in the environment too, your answer is that these genotypes, red and blue, don't really regulate your phenotype, right? So you go check, there's nothing going on, you forget about it. But if you are the lucky student who happen to choose environment 1 to run exactly the same experiment, now you see that red and blue actually determine a lot of the variation in your phenotype. And this is what we call cryptic genetic variation because the variation exists in your population, but is hidden or cryptic depending on the environment when you measure the genetic effect, right? So it's just a toy example of what cryptic genetic variation is. And we think that's what's going on here, where we have the genetic variation, as I told you, is the same genetic pool. And now these genetic effects are actually relevant when you are in the right environment, which is high sugar. Okay, I will finish with this. Christian has seen a very similar talk recently, and some of you guys who were in Paris. So I decided to add something new so you don't get bored. The question here, as I told you at the beginning, is that the GP map is not just gene expression to genotypes or morphology to genotypes. The GP map is this complex, hierarchical structure where information propagates from the genotype to many phenotypes, right? So here we're asking, okay, we measured this. I told you the stories about gene expression variation, blah, 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 blah. Does this insanity that is going on at the gene expression level propagate to other layers of the genotype-phenotype? So this is a project that Saudat, a PhD student in the lab, has been leading for her PhD, plus an army of people who are Hibis, Erasmus students. Anyone who goes through my lab will contribute to this experiment just because it's just very painful to collect this data. And what Saudat did is she used the same population as James used, exposed it to control and to high sugar conditions, exactly the same setup, are collected around 1,000 flies, and for each individual fly, she measured all this. First, she measured function so she measured flight performance, and I talked to some of you guys before about that. Flight performance of each individual fly as a proxy for function. Then she dissected the wings of these flies and used the shape of these wings as a proxy for morphology. But because she likes to have a really tough life, she collected left and right wings, because as I told you at the beginning, if you compare right and left sides, you can approximate developmental noise. So we are using this metric as a proxy for developmental processes going on at the individual level. And then, of course, because of GPMAP stories, then we collect genotypes to try to do this mapping. So I'm just going to give you a snippet of what's going on here in this preliminary data. Is when you think about the first layer of development, where we are using developmental robustness, right and left proxies, she finds that the high sugar flies are more asymmetric than the control flies. So more asymmetry means more noise in your developmental process. So in this sense, these flies that have all this noise in their transcriptome also are noisier in the developmental process. When we check morphology, using, as I mentioned, the shape of your wings, this is a PCA of shape. Shape is just a multivariate vector of X and Y coordinates of these little dots here who are called landmarks. So this is a principal component of each individual fly shape vector. You can see in gray is the 95% confidence of the distribution of your control shape. And in orange is the distribution of your high sugar flies. So I hope you can see that the variance or the phenotypic space that these wings can achieve in high sugar is wider than the phenotypic space that you can explore in control conditions. And then we get to function, which is approximated by this assay that was developed by a different lab. And here, there's no difference. Flight performance of high sugar and control is the same, and the variance in the individual values is also not significantly different. I told you when you get to fitness, I showed you this plot before, the egg to adult survival ratio of high sugar and control flies is pretty much the same, right? So this is just a kind of a quick summary of some of the phenotypes we're collecting. And then so far, what we believe is that this increase in expression variability propagates to some layers of the GP map, but at some point it gets buffered and it doesn't really reach other layers of the GP map. And we could argue why is this the case. So we have increase in variance, variability or noise at the transcription level. We have increase in asymmetry or developmental robust. So they become less robust in high sugar in development. They increase variance in phenotypic space. But when you get to the higher layers of this GP map, then there's not really change, right? I will leave that there so you think about it. I don't know what else to tell you. This is very preliminary data, but we're very excited because we could argue what was your expectation. Does it propagate? Does it buffer? Where does it buffer? What does this mean? Is the same GP map true for every single trait or not? We haven't done that, but we will. And I will let you know next time we see you. Okay, so with this I will finish. I just want to highlight 3 things about gene expression variability. First, it seems to have been shaped by evolutionary forces in different species. It responds to environmental perturbation in a way that we believe is biologically important. And it seems to be genetically regulated. There's plenty of genetic variation underlying expression variants. Now the $1 million question is, how does it relate to higher order phenotypes? Right? Like, can I actually connect genotype to transcription to other phenotypes? I show you a snippet of the results, but we'll see where we get with that. And lastly, how does variability evolve? Everything I told you before is one generation, could be irrelevant. We don't know. So what we are doing now is conducting experimental evolution on this high sugar setup. And what we hope to see is first what happens with variance in the dynamics of adaptation, and what happens at the end. I don't know, what are your thoughts? Is variance going to go back to normal? Is it going to remain high? I have no idea. Also, because we have the SNPs that regulate variance, we will be able to track what happens with the frequencies of high variance alleles or low variance alleles. Talk to me in the future, and I hope to have answers for all this stuff. With this, I will finish. Thank you very much for the invitation. This is my amazing lab up here. Saudat and James, who contributed to the stories I told you today, collaborators, all the guys who have gone through the lab collecting fly wings for us, and funding, of course, and I'm happy to take any questions.